Chapter 17 is going to look at gene expression. So this is how our genes, or our DNA, get expressed in the form of proteins, which are what make you look the way you look and work the way you work. So this is going to happen in two main steps. We're going to go from DNA to RNA, and this process is called transcription. It's also going to include something called RNA processing to make the RNA ready to leave the nucleus. And that RNA is going to be translated into proteins. This is called translation. So we're going to have three main processes here: transcription, RNA processing, and translation, that we're going to need to go through to get from DNA all the way to proteins. So the first understanding of how genes and proteins were related started out as what was called the one gene, one enzyme hypothesis. So we know that molecules are produced in metabolic pathways, where we have one molecule and then there's an enzyme that turns it into another molecule, another enzyme that turns it into another molecule, and so on. So here's my metabolic pathway. This one has a precursor, and then three reactions that eventually result in the product, which here is arginine. So Beadle and Tatum were the first ones to track mutant genes, so genetic mutations in the genes that code for these three enzymes, and then find mutant classes of mold that were not able to survive based on what enzymes were broken and which precursors they were given. So when they did this experiment, the wild type, which again is the common type, which would be the ones with no mutation, if they were given the precursor, they were able to survive because they had all three metabolic pathways intact, and they were able to go ahead and produce arginine. Arginine is an amino acid that we need to be able to produce to survive. And then they found three different classes of mutations that were able to survive in certain situations because some of their genes were intact, but some of them were not. So class 1 mutants had a mutation in the gene for enzyme A, which means that they were not able to produce enzyme A. So the protein synthesis going from gene A to enzyme A got disrupted. So given the precursor, those organisms did not survive, but if you gave them the second molecule, ornithine, they still had B and C, and they were able to survive and make arginine. Class 2 mutants had a mutation in the gene B, which coded for enzyme B. So unless they were given citrulline, they were not able to survive. But if they were given citrulline, they did have enzyme C, and they were able to survive. And then the last mutant class had an enzyme C mutation in gene C. So gene C was not able to produce enzyme C. And no matter what you gave these organisms, they didn't survive unless you gave them straight up arginine, because they couldn't do this metabolic pathway at all. So this was the first kind of experiment where we realized that a specific gene was related to a specific protein, and if the gene was broken, then the function of that protein was broken. Eventually, we went on to realize that not all proteins are enzymes. Enzymes are proteins, but not every protein we have is enzymes, so we kind of renamed this the one gene, one protein hypothesis. And then we realized that many proteins are composed of many polypeptides. So for example, hemoglobin has 4 polypeptides, 2 alpha and 2 beta, and each of those polypeptides is different, so they need their own gene. The most recent hypothesis now is the one gene, one polypeptide hypothesis, where each distinct gene can code for a distinct chain of protein, which is a polypeptide or a polymer. So RNA functions as the bridge between our DNA, which is our genetic code, and the proteins, which is our expression of that genetic code. And we can't go directly there. What we need is we need to go to RNA first and then go through this expression. And we believe that this is actually because RNA evolved before DNA in the first organisms that lived on this planet. So the first couple of steps here, we have transcription. And this is the synthesis of RNA from DNA. So we would take DNA and complementary base pair to make what's called an RNA transcript. The finishing of this RNA transcript is going to require something called RNA processing, so that it's going to be safe to use this RNA out in the cytoplasm. And then the third step is going to be translation, which is where we're going to take this messenger RNA and translate it into a polypeptide. And we're going to literally make a chain of amino acids that will then get folded into the correct shape. And we know that this happens in the ribosomes. Those are the organelles that are responsible for making proteins, so protein synthesis. This is going to look a little bit different in a bacterial cell versus a eukaryotic cell. A bacterial cell does not have a nucleus. So the DNA is just in the cytoplasm. There's no physical barrier between the DNA and the ribosomes. We don't actually have to do RNA processing. In a bacterial cell, we only need the transcription to go from DNA to mRNA. That mRNA doesn't need to be changed. And then the second step would be translation, which can happen physically right there at the ribosomes. In a eukaryotic cell, because we do have a nucleus, the DNA is contained inside the nucleus. It needs to be copied into RNA to be able to leave the nucleus. So we're going to do transcription to copy DNA into what's called pre-mRNA, or the RNA transcript. The second step will be changing this mRNA into mature mRNA that is going to be able to leave the nucleus into the cytoplasm. And then the cytoplasm is going to be the place for translation, which is where we're going to read that mRNA using ribosomes into a protein. So this is what's called the central dogma of biology: DNA to RNA to protein. So you should be dreaming about this in your sleep. You should have this down pat. DNA to RNA to protein. DNA to RNA is going to involve transcription to make pre-mRNA, and then RNA processing in eukaryotes to take that pre-mRNA and turn it into messenger RNA. And then RNA to protein is a process called translation, which is going to happen in the cytoplasm at the ribosomes. So we know that the genetic code in the DNA contains 4 nucleic acids. We've got adenine, thymine, guanine, and cytosine. Only 4 nucleic bases. However, there are 20 different amino acids. So in order to code for 20 different amino acids, we need to group some of these letters together. We can't do a one-to-one code. So if the DNA code was only 1 base letter long, we would only be able to code for 4 amino acids. We would have 1 for A, 1 for T, 1 for G, 1 for C. That wouldn't work out well because there's 20 that we need to be able to code for. If the DNA code was 2 bases long, you can do all combinations of these, A's, T's, G's, and C's, and eventually get to, so all the different permutations of 4 letters with a sample of 2, you only get 12 different amino acids. And that's more, but it's not enough. It's not 20. So 12 amino acids is not going to give us enough codes to be able to code for all 20 of our amino acids. So our DNA has actually settled on a triplet code. So you have 3 nucleic acids in a row, and that is a codon. With that, we can code for up to 64 different amino acids, which, while 64 is more than we need, will cover the 20 different amino acids that we need to code for. So this is our triplet code, or codon. These are read in the 5' to 3' direction. So the way this works is in the DNA, we have one of the 2 sides of the DNA double strand is the template strand. So the template strand will run 3' to 5', and then the messenger RNA will be made in the 5' to 3' direction. For any one gene, the same side is always the template strand. So in this example, this top strand here is the template strand. For a different gene, the bottom strand might be the template strand. The template strand just depends on which actual protein we're going to make, because it might be different depending on which strand we use. So here's my template strand of DNA, and it reads A, C, C, A, C, C, G, A, G, T. So what would happen is we would go ahead and break this double strand apart, And we would focus here on just the template strand for transcription. And in order to transcribe this DNA into RNA, we need to use complementary base pairing. So when we have DNA and RNA, a T in a DNA is going to get coded with an A in an RNA. Just like complementary DNA pairing, A always pairs with T. G pairs with C. C pairs with G. The only one that's different is that when you have an A in the DNA, because RNA does not have thymine, RNA is going to instead insert a U for uracil. So you can see here that we have made the complementary base pair, U to pair with A, G pairs with C, G pairs with C, U pairs with A, G, C pairs with C, G, and UCA pairs with AGT. So these are complementary to the template strand. And then the last step is this translation, where we're going to read these triplet codons into amino acids. One thing that's really amazing is that the genetic code of which codon codes for which amino acid is the same for all organisms on this planet. So all organisms that use RNA to proteins use the same code that we use. So it's just proteins in our body that read the codon and insert the correct amino acid. And we have painstakingly figured out the code for the 64 different possible triplets to code for 20 different amino acids. So this is what we call a codon chart. So the first amino acid is over here. The second is at the top, and then the third is at the right. So if I had the code UCG, the first one is over here, I would start with U. C is my second one, so that means I'm in this box here. And then G is my third one, that's right there. So UCG is right here, and that's going to code for this amino acid serine. The other thing you can do is just kind of search the chart for the right one, but by using the boxes on the side, you can find what you need faster. There is one start codon, AUG, which codes for methionine. This is also found at the start of every code, so this is where we're always going to start translation. I like to remember this because AUG is the abbreviation for August, and we always start school in August. You are not going to have to memorize these. I am going to give you this table anytime that you need to use this table. There are also 3 stop codons. So there are 3 codons that tell translation to stop, UAA, UAG, and UGA. Anytime a ribosome reaches these 3 codons, it will stop translation and release the amino acid. So let's look at a couple of these words. The genetic code is redundant. Redundancy is when you have kind of backups for backups. So you can see that there are 2 different codons here that code for leucine, UUA and UUG. So those 2 are redundant. They both code for the same amino acid. However, it is not ambiguous. Ambiguous is when you don't know if it's one or the other. So every single triplet code has a very specific one amino acid that it codes for. There are no triplet codes that code for arginine or leucine. Arginine has its own codes, leucine has its own codes. There's some redundancy because there's 4 different, there's 4 here, 2 here that code for arginine. So there's 6 different ways to get arginine, but all of those 6 ways will specifically give you arginine and nothing else. So it is redundant, but not ambiguous. So let's try this. We'll do a practice. This is what you're going to be doing in your protein synthesis assignment. You need to be able to go from DNA to RNA and then use a codon chart to read the amino acid. So we start with the DNA and complementary base pair that to make RNA. And I like to leave the little dashes to make sure the codons are easy to read. So T will pair with A pairs with U, C pairs with G. And then we can keep going. GAT is going to be CUA. CAG will code with GUC. CGT would code with GCA. TGC will code with ACG. and then ACT will code with UGA. So those are my messenger RNA codons. And then we would use our codon chart here to translate this into amino acids. So AUG is met or start, so I can say met. CUA, so CUA is leucine, LEU. GUC, GUC right here is valine. GCA, GCA is alanine. ACG, we've got ACG is threonine. And then UGA is UGA. That's a stop, so I'm just going to write stop. If there were more codons after this, I wouldn't continue to read them. Once you reach a stop, you stop translating. You don't continue to translate past that. At that point, the ribosome would stop. So you can see that we can complementary base pair A's to T's, U's to A's, G to C, C to G. And then we can use these 3 at a time on the codon chart to be able to read them into the amino acids that would then make up, this is the polypeptide.